This is an A-level biology video about the structure and properties of some key carbohydrates, including how monosaccharides join together to form polysaccharides. I went through this in more detail in my video about monosaccharides and disaccharides, but just to recap, carbohydrates are molecules made from carbon, hydrogen and oxygen only, with the general formula CxH2O-Y, where X and Y can be the same number but they can also be different numbers. Carbohydrates are also known as saccharides, and therefore we can classify them based on their size into monosaccharides like glucose, fructose, galactose and ribose, disaccharides like lactose and sucrose, which contain 2 sugar units, and then polysaccharides like glycogen, starch and cellulose, which are very long polymer chains containing many monosaccharide units. In this video, we're primarily interested in complex carbohydrates, which have roles both in the storage of energy and also in structural roles. In addition to the 5 monosaccharides and 3 disaccharides that you need to know about, there are 4 polysaccharides, or 5 if you count the 2 different forms of starch. So we have starch, which is found in the stroma of the chloroplasts and also a bit in the cytoplasm, and this is made from glucose. Then we have glycogen, which is found in animals, in both muscle cells and the liver, and this is made from alpha glucose only. Cellulose is found in the plant cell walls, and this is made from beta glucose. So remember from the previous video, the difference between that alpha glucose and beta glucose is the position of the hydrogen and the hydroxy groups on carbon number 1. And then chitin is the polysaccharide that makes up the exoskeleton of arthropods, and this is made from a monosaccharide called glucosamine. 3 of these polysaccharides are based on glucose, so it's important that you're familiar both with the structure of glucose and with the difference between alpha glucose and beta glucose, which, as we've just said, is the position of those hydrogen and hydroxy groups on carbon number 1. Monosaccharides like glucose can be joined together by a chemical reaction called a condensation reaction, in which 2 smaller subunits are joined together with the loss of a molecule of water. When we're talking about the formation of carbohydrates, this forms a specialized covalent bond called a glycosidic bond. 2 glucose molecules can be joined together by a glycosidic bond to form the disaccharide maltose. But if we carry on adding more glucose, then eventually we can make a larger polysaccharide. Similarly, in the reverse of condensation, hydrolysis is a process where water is added to break the glycosidic bond and retrieve those original monosaccharide monomers, which can then be used for respiration and also for making other molecules. Excess glucose that was produced during photosynthesis, but that isn't immediately needed for respiration or to produce other molecules, is stored as starch. So this is the primary storage polysaccharide for plants. Whereas at GCSE we just talked about starch, now you need to learn about the 2 different molecules that make it up. So these are called amylose or amylose and amylopectin. The first of the 2 polysaccharides that make up starch is amylose or amylose, and this is a very long chain of alpha glucose molecules. These only contain 1 to 4 glycosidic bonds between them, so these are the exact same glycosidic bonds that we drew when we were making a molecule of maltose, but whereas maltose only contains 2 glucose molecules, amylose contains several hundred. It forms this very, very long chain that has a characteristic twisted shape. It can be very compact, and this makes it much less soluble than the glucose it's made from. This is important because if the plant cell kept the glucose without turning it into amylose or amylopectin first, then this would draw in a lot of water from the surroundings, and this would not be good for the cell. It's important to remember that because amylose doesn't have any branching, it can only be broken down from either end of the molecule, and this means that breaking down amylose is going to be a very slow process. In contrast to amylose, which is just a long straight chain, amylopectin is branched. So approximately every 25 glucose molecules, there is a 1 to 6 glycosidic bond as opposed to a 1 to 4 glycosidic bond, and that leads to another branch coming off the main chain. Just like amylose, it is insoluble, but because it has this branched structure, it's much faster to break it down or build it up again because the glucose monomers can be broken off at a number of different points at the same time. Glycogen is the storage polysaccharide that we find in animal cells, and it's very similar in structure to a myelopectin in that it's made from alpha glucose that is primarily joined by these 1 to 4 glycosidic bonds, but it also has this branched structure. But actually, glycogen is even more heavily branched than a myelopectin is, and this makes sense if you think about what the advantage of being branched is. As we said, it's much faster to break down a myelopectin than amylose because it has all that extra branching. So this means that you can get energy faster. And if you think about it, it's much more likely that an animal cell, which is mobile, is going to need a sudden influx of energy compared to a plant cell. So therefore, it makes more sense to have more branching. Glycogen is also very compact, so it's good for storage. Glucose monomers can be added onto the glycogen molecule by enzymes and also removed by other enzymes. Cellulose is the odd one out of our polysaccharides in that it's made from beta-glucose, not alpha-glucose. So remember, the difference between the two of those is the position of the hydrogen and the hydroxy group on carbon number 1. Now, in order for these to bond together, alternate molecules need to be turned upside down. So basically we have one glucose the way up we would normally draw it, and then the other one flipped over. Obviously, if you were drawing this in an exam, you would have your letters the right way round, but this is just me having flipped my diagram over. So cellulose forms a very straight chain molecule. It doesn't coil like amylose, and it doesn't form branches like a myelopectin or glycogen. But what this means is that there can be hydrogen bonds between the chains of cellulose, and these form microfibrils. And these are really, really important because they give structural support to cell walls. So you can see here we've got the hydrogen bonds between the layers, and this whole structure together is called a microfibril. So pause the video and explain, how does the structure of cellulose relate to its function? Well, firstly, it is a straight chain molecule. It doesn't curl, it doesn't form a helix, and it's also not branched. This is important because it allows many hydrogen bonds to form between the chains, and this confers strength. Now is a good opportunity to summarise what you've learnt in this session. So pause the video and see whether you can fill in this table. Amylose and a myelopectin are energy stores in plants, whereas glycogen is an energy store in animals, and cellulose is a structural polysaccharide used in cell walls. Amylose, myelopectin and glycogen are formed from alpha-glucose, whereas cellulose is formed from beta-glucose. Amylose only contains 1 to 4 glycosidic bonds, whereas a myelopectin and glycogen contain 1 to 6 bonds as well, and cellulose contains 1 to 4 bonds. The overall structure of amylose is a helix, whereas a myelopectin is branched and slightly coiled, as is glycogen, and cellulose forms straight chain molecules. All 4 of these polysaccharides are insoluble due to their large size. Thank you very much for watching, and I hope you're now feeling fully confident in your knowledge about polysaccharides. If you did find this video useful, then don't forget to like, subscribe, comment and share, and I'll be back soon with some more A-level biology videos.